Clearly, I just can't be introduced. So as Aaron alluded to, I am an assistant professor in the Department of Geoscience, which is in the College of Science at the University of Arizona. And I am the director of marine research at the Biosphere too. So I work closely with Katie and our team to transform the Biosphere 2 ocean into the state-of-the-art reef lab to really try to develop solutions for coral reefs under a changing climate. And as I'm sure we'll get to today, my own research program focuses on using corals to really better understand how oceans have changed in the past so that we can better predict how oceans will continue to change and in turn help buy some corals some time to ensure that we have reefs in the future.
Great. And I guess what I'd like to start with in honor of Earth Day with a very simple question: what is a coral?
It's a great question, Aaron. It's often the first question that we get, in part because it's a bit confusing, a little bit misleading. So in a lot of ways, corals, although they are animals, have a lot of rock-like and plant-like features. So here's a picture of what a coral organism looks like, a couple of individual, what we call polyps, the individual coral organisms. So these are really closely related to jellyfish and sea anemones, or anemones if you're Nemo. And so they are animals, and they do feed with these tentacles that you can see here. So they have sting cells that actually capture prey and bring them into its mouth. But actually, a lot like plants, they also have these algae that live in their tissue, and that's what's shown here. And these small, what we call symbiotic algae, so a symbiosis means a relationship that's mutually beneficial. These algae provide actually the majority of the coral's nutrients, their energy. And so these are really critical for the health of the coral organism. And in turn, the corals provide a safe home and waste products that actually help with their ability to produce energy. And then finally, what we know of coral reefs is they actually have these great rock-like features. So just like the shells that you find on the beach, corals produce a skeleton that's very similar to that same mineral that you find on the beach. And produce these massive structures that are so critical for providing habitat for a diversity of organisms, and ultimately even protect our coastlines from storms and growingly sea level rise. So corals are complicated. They're animals, but they are also in many ways rocks and plants as well.
Yeah, and maybe as a segue into a bit of the history of the Biosphere 2 ocean, I'd like to start by asking, this biosphere has a legacy. It's these systems that we're studying now are about 30 years old. And with that, this ocean has reached some sort of equilibrium state. And when did you guys decide to start to use this system for coral research specifically? And how is its legacy as a tropical coral reef in the past and now soon to be again? How does that kind of tie into this narrative?
Yeah, absolutely. I'll start that off and I'll actually pass it over to Katie, because a lot of that sort of seminal work that sort of brought us to where we are today started before I arrived on the scene. But I do want to show one thing that'll sort of set the stage for what Katie will talk about. And that's that, prior to this experiment, over sort of decades of collective coral reef research, we've sort of learned what we call sort of the building blocks of a resilient coral reef. And resilient coral reefs are reefs that can withstand changing ocean conditions and persist in the face of climate change. And so we'll get to coral reef health more in detail sure later, but we have learned what these key building blocks are, how algae play a role, how various reefs around the world fare. And Dr. Julia Cole and Dr. Ruth Gates, Katie, and a number of others around the coral reef community came together and realized that we have a really unique opportunity here at the Biosphere 2 to move from this decades of work on process, so what's happening and why it's happening, to really apply this and develop these solutions. So how can we build a resilient reef that will persist through changing conditions and exist for our children and our children's children? And so I want to pass it over to Katie to give a little bit more of that backstory. But really, it was I'm standing on the shoulder of those giants who had this vision for the project.
Yeah, so the Biosphere 2 ocean has an incredibly unique history. So the system behind me here is a 1 million gallon tank of salt water. And in the early 90s, you heard, if you were tuning into our last discussion with Jane and Tabor, they discussed actually going out to the Yucatan Peninsula to collect corals and fish and all kinds of animals to put into this reef system. So this ocean in the past has been a diverse tropical coral reef and survived at, inside not only the biosphere here, but at 4,000 feet elevation. So under a lot of different circumstances than they might see in the natural systems. And that reef was able to survive in here. So we know that's, data point number one for us is that we know that corals survive in this system behind us. And there's been incredible seminal studies that have come out of the Biosphere 2 ocean done by Columbia University in 1996. They really were looking at the effects of increased CO2 in our atmosphere and how that was affecting coral reefs around the world. So as CO2 goes up, we see our oceans really take up that CO2, and that negatively affects the calcification rate of these corals. So their ability to take ions that they build their skeleton with, they can't do it as efficiently with that increased carbon dioxide in the atmosphere. So we see them start to become stressed, and we were able to observe that right here in this system. We were able to see that stress response, which is called coral bleaching. You might have heard that term before. It's when the coral turns that stark white. So we saw our corals in the system under a lot of stress. Diane, so yeah, we've got a screen up here. So that middle section is that bleaching. So that's kind of what those corals look like when they're under that kind of stress. And after that experiment, we had seen, these corals be stressed, and over time and over different management, those corals went through different things, and we now today don't have any living corals inside of the Biosphere 2 ocean. So we've had over the years, over the last couple years, we've tried to decide what is the best way to use this incredible system that we have precise control over to be able to have an impact. And like Diane mentioned, Dr. Julie Cole and Dr. Ruth Gates were kind of the first people to come together and decide that this system can really be used to identify solutions that we can then put back into the natural systems to help save our coral reefs around the world.
Thank you, Katie. And some of our first questions coming through have to do with specifics of the ocean itself. Do you guys have a tidal system there? What kind of animals are in the water? So perhaps one of you or both of you could kind of give a general description of the Biosphere 2 ocean. How deep is it? What are some of the support systems that go along with it?
Yeah, I'd be happy to start that off, and I can pass it off to Katie as well along the way. So just to take a little bit of a step back here. So, we've both talked about how this system is so unique to test these solutions before they're applied in the wild. And that is because we have this sort of Goldilocks, if you will, of scale and control. And so fundamentally what makes the Biosphere 2 one of a kind for testing these solutions is that we both can test them at what we call ecosystem scale. And by that I mean we understand how these solutions play out from microbes, these tiny little organisms that we can't see, all the way up the trophic food web to the top sort of consumers, and everything in between. And so we really need to understand the system as a whole in order to understand which solutions will work in reality on the real world reefs. But the second piece of the problem is control. So we still need to be able to, at ecosystem scale, test very realistic conditions that we might see in the future. And it's often hard to pull apart these various drivers of stress that Katie alluded to, temperature and ocean acidification, so the decrease in pH of our world's oceans. How do we pull those apart and know what's driving, who did it, if you will? And so we have this Goldilocks of both scales, 700,000 gallons, but yet very precise control over the system. So coming to the question from Kara about tidal fluctuations, that's a great question, and one of the examples of control. So we don't have tides per se in the system, but what we do have on the far end of this system that you might be able to see here, and those of you who are in the previous webinar with Clay and Tabor, you saw, or Clay and Jane, sorry, you saw that they talked about the wave wall and the sound that it makes as they're on the beach. So this is a wave wall that produces realistic waves that would happen on a coral reef ecosystem. So that's one example of how we realistically represent a coral reef. Want to show you one other thing. So just to wrap that up, I want to sort of dive a little bit into the nitty-gritty, but I will walk through this figure. But one of the things that the creators of the Biosphere 2 did a really good job of, in addition to getting waves and realistic circulation on the reef, is they actually mimicked the bathymetry, the shape of the ocean, that looks a lot like a real coral reef. So those of you who have been to or seen images of the Great Barrier Reef and other barrier reef systems know that there are these sort of structures that line the world's coasts that look like what I showed earlier. So that's exactly what the shape of the Biosphere 2 ocean is. We have this shallow reef crest, a backloop reef lagoon, and a deep fore reef environment. And so this is actually really, really representative of what we see in the real world. So this back reef lagoon, it's a shallow reef crest, and then this deeper fore reef that goes to about 7 meters depth. So all that to say, we have this great scale, so 700,000 gallons. It's realistic, and we can very precisely control it.
And maybe pivoting on that to Katie, we've had a couple of questions as to what animals do you have in the system right now?
Yeah, it's a great question, and one we get a lot out here. When you think about the ocean, you automatically associate those animals with it. And we've been adding animals to our system recently. In the last year, we've really pushed to add organisms to the system. And the main reason right now that we've chosen certain animals is that they're herbivores, so that they eat algae. Our system, after those corals over time died off in the system behind me here, we got a lot of algae blooming, and that's pretty typical. It's what you see when coral reefs die off in the natural systems. You tend to see algae take over. So we have the same kind of reaction here. So we've been removing this algae pretty heavily in the last year. So we've removed about 10,000 pounds by hand over time, and that involves us going in and diving and putting them on these trays and sloshing it onto the beach and then taking it out of Biosphere. So it's a lot of manual labor. We get a good workout in those days. But we've also recruited some animals to help us keep that algae at a balance. And that's really important. We can't do all the hard work in here. We need something to kind of help us eat that algae as well. So I actually have some of my friends next to me here. So let me pull out one of my favorite animals that we've added. So this here is a short spine sea urchin. On top of him, he collects these little shells to help camouflage him here. But these guys we introduced last April, and they're amazing algae eaters. So they get in here and they help just kind of clean off those rocks. So these are one of the key animals that we've added. We've added them last April, and then we added more this past February. So when we go in here, we dive, we see them every time we go in, all on top of those rocks, especially on the reef area. They love it over there. One of the other a little bit smaller animals that we've added, but still equally as important, are these hermit crabs. I don't know if you can see him. He might be kind of hiding, he's camera shy. But these guys are also really great algae eaters. We've added thousands and thousands of these over the years, and they just really help us keep that algae at bay. Now we also have about 7 species of fish in here, and some of those fish are descendants from the original mission or from Columbia's era. So last year in April, we actually added the first fish since 1996, and this was really, really exciting for us. We knew at this point our system was healthy enough to add animals like this back in. So you can see up on the screen at the bottom there, you can see the ocean surgeonfish. That is one of the animals. We added 22 of those guys, and then we had the red-banded parrotfish. Those ones, we added 2 of those, and we plan to continue to add more species of fish as we move forward in this project to really help us keep that algae under control. A healthy balance of algae is good in a reef system. You just don't want it taking over the corals.
Thank you, guys. That gives us a really rich representation, both kind of structurally and from the perspective of what animals we have in the system. I want to move our gaze outward a little bit to kind of create a foundation for why we're doing research. We had one question from Kat about how long do we have before coral reef damage is considered beyond repair? And what are some of the more root causes of the need for research on corals in an experimental system?
Yeah, I mean, this is a great question. This is the core of why we're here and doing what we're doing. And so I want to give just a personal anecdote. I'm here because I personally have a tremendous amount of hope. So I'm newest to the project here. I've been here for about 2 years now. I came from Boston University, where I was before that. And when I heard about this opportunity to do coral reef research in the middle of the Sonoran Desert, I did not hesitate to apply because, like I said, we need to be thinking about solutions. But on a personal level, I've gotten here because I have not been without my share of seeing corals that I have grown to love completely decimated with local pollution and stress, as well as these major global drivers. So these are some examples from some field sites that I work on. These particular reefs are in the Marshall Islands, which is in the western tropical Pacific. A very small island nation that is going to be really heavily impacted by climate change and rising sea levels. So having seen reefs impacted by trash and covered in algae, following some of these really big stressors that Katie was talking about earlier, I'm very motivated to find solutions. But the other thing that I know is that we have seen evidence in the past that coral reefs can recover. The challenge is that we need to buy them time. So to sort of get to the answer to your question, one of the biggest challenges is that these bleaching events that Katie talked about are happening more and more frequently and more intense because of warming of our world's oceans. And when you combine it with the fact that as we release CO2, fossil fuels into the atmosphere, the oceans are actually taking up a large majority of that carbon dioxide. And with that, just like the CO2 that's pumped into our Coke bottles, our ocean is becoming more and more acidic. And this is really detrimental for coral reefs. So to answer your question, I've seen recovery on reefs. I have a tremendous amount of hope. We have colleagues all over the world who have come up with really unique solutions that I'm happy to talk about here in a little bit in more detail. And so we need to do two things. We need to buy corals time by addressing climate change, the elephant in the room. In the meantime, our team of 50 scientists from all over the world, we're going to come up with the best concoction of various solutions are to build the most resilient reefs we can all over the world.
And Katie, perhaps just briefly, you could chime in about your recent trip to the Great Barrier Reef in Australia.
Yeah, so I had the privilege to go to the Great Barrier Reef in February, and February in Australia is their kind of peak of their summer months. And we got to go out on the Great Barrier Reef. I hadn't ever gone before, so it was, you know, a dream of mine to go out and see this reef, and it was as incredible as I thought it might be. It was so beautiful and so diverse, but as we are looking around at the corals in front of us, we also noticed what was underneath those alive corals was skeletons of corals that have been taken out in the very recent years, in the last 3 years, by these bleaching events. So for me, it was not only humbling, but also very motivating to see that, you know, there is life on top of corals that have died because of these different stressors, but they were still starting to get stressed out as well. So we, as a coral reef community, coming together and identifying solutions so that we can buy these corals time. You know, if they are continued every year to be hit by these massive heat increases and they have to deal with that every year, we're just hoping to buy them some time so that we can continue to have reefs in the future.
Now, the current state of our Biosphere 2 ocean is a bit of a degraded one. And we say that because it had a fair amount of algae covering most of the surfaces. And while we've extracted a lot of that algae out by hand, and we have herbivorous fish and invertebrates working for us biologically, how is the degraded state of the Biosphere 2 ocean actually an advantage in starting to plan out research for testing resilient features of coral reefs?
That's a great question, and that's really at the core of what we've been doing over the last couple of years is we're re-engineering and updating the life support systems. So for those of you who haven't seen the Biosphere 2 ocean in the last few years, just to put a bit of context behind what Katie was talking about, this is a 3D map, basically of stitched together photos that our collaborators at Scripps put together of what the Biosphere 2 ocean bottom looked like in 2018. So really every surface of the bottom of the ocean was covered in this algae that Katie described. So now this algae obviously has been removed. As she said, about 10,000 pounds of algae was removed by the team over the last year. But during that period, over the last couple of years, we used it as an opportunity to test what it takes to go from a degraded reef system like this one, which fortunately we haven't seen reefs that have been this degraded for over much of the world's reefs. But this could be the future of the reef if we continue to do nothing. So we had the opportunity to say, okay, what happens when we have a degraded system like this? How can we rebuild a healthy coral reef? So what we did is we spent the last couple years studying everything that we could about this degraded reef system. Studying the physical environment, so what sort of environmental conditions, the temperature, the pH, the chemistry of the ocean, and of course what the biology was doing. So everything from these tiny little microorganisms all the way up to the fish that Katie described. So we're really trying to understand natural cycling on this degraded reef and test what we call bioremediation solutions. So as it sounds, bio, organisms, so natural solutions for remediating or shifting that balance back towards a healthy reef. So we've learned a lot. In a couple examples shown here, we actually took a core. This is me and my PhD student, Alice Chapman. And this core of the sediments that accumulated in the Biosphere 2 since that original experiment that Katie described in the 90s, we have decades of sediment that's accumulated. And what we learned is we can look at the chemistry of that sediment and know why the system has shifted from this healthy ecosystem towards an algae-dominated system. And then, of course, an example here on the bottom of some of the biology. These are some of the key, what we call phytoplankton, so the plants that actually produce the base of the food web here in the Biosphere 2. And this is work that's being led by Dr. Jeremiah Hackett from the University of Arizona.
Thank you. Diane, I'm really curious personally, and I think others are, about your background in geosciences and the way that you don't just study coral reefs, but you kind of deconstruct sediments of past oceans in some ways, right? Mm-hmm. Can you speak to how, to maybe some of that research that you do, but also how that plays into the biosphere as a kind of a proxy system for understanding these longer timelines of our ecosystems?
It's a fantastic question. And one I get asked a lot, and one that I also think about a lot myself, because just to take a really big step back, this is a compilation of pictures of me as a kid when I first saw a coral reef for the first time on the left on a family vacation to Buck Island National Monument in the Caribbean. To the right, where I'm now drilling holes in corals to study climate change. And if you would have asked myself as a kid, what I would be doing in the future, I can tell you it wouldn't be poking holes in corals to study climate change. So how did I get here, and how do we use this to develop solutions? Well, the short story is, on these trips, I was fortunate to see a healthy coral reef ecosystem, and then it was pristine, it was protected. But unfortunately, I returned in college quite a number of years later as an intern for the National Marine Fisheries Service studying coral reefs, and jumped in the water, and my snorkel fell out of my mouth because the reef was completely gone. And so for the first time as a young adult, I saw firsthand what can happen to a coral reef because of climate change. So to answer your question, from there, my career took a hard right turn or left turn, whichever way, from coral ecology and biology to geosciences, because we can actually use corals to better understand these global scale drivers of stress on coral reefs. So now what we do, and this is an example from our work in the Marshall Islands, is we find these giant corals. Some of these can be the size of like a VW bus, either living, or in this case, a fossil colony, so one that was dead already when we cored it. And much like trees, they grow in these annual growth bands. So you can see right here by eye. And so like tree ring research, we can use these rings of growth to look at what the ocean conditions were in that snapshot of time during which that skeleton was emplaced by that coral. So we get this rich history of ocean conditions over hundreds of years when individual corals lived. And so to bring it back to the Biosphere 2 then, what this work has shown us is that, first of all, coming back to my point about hope, is I've taken cores out of colonies like this and seen evidence that they died. Got covered with algae, covered with bivalves and other reef organisms. And then a couple years later, they pick back up and they start regrowing. And then sometime later, it happens again. So this evidence for regrowth and evidence of hope. But in terms of what we're doing at the Biosphere 2, one of the things that I learned from this work is that corals in regions that are hit really hard by stressors, so they are frequently hit by extreme events, they tend to do better than corals that are in very calm, stable environments. And so when I said earlier that we've learned about the key building blocks of coral resilience, that's this top building block, is we know that corals here in this part of the Pacific in red that get hit really hard do better than the reefs further west. And that's a really key piece of information, because fast forward 10, 20 years, we now use this as a solution. So really smart colleagues have developed what we call stress hardening. So we harden corals, we turn up the temperature, we stress them out, and then we transplant them onto the reef. And they're more likely to be resistant to these stress events in the future. And we know this because of work where we core corals to look at past events. And so that's how it kind of all fits together and why we're doing what we're doing.
So is stress hardening something that you guys can do in the Biosphere 2 ocean? And what other forms of kind of resilient experimentation are you looking into to supplement stress hardening?
Great question. So absolutely. So that's going to be one of the main things that we test here, and among other solutions I'll talk about here in a second. And the way we'll do this is actually capitalize on what we call an experimental raceway system. So this is a 2,700 liter or 270 gallon tank, so much, much smaller than the 700,000 gallons in the Biosphere 2. And this is a separate system where you can see it has its own life support. So it has its own control to turn up the temperature. And we can use this system to harden the corals under different climate change scenarios and transplant them into the Biosphere 2 ocean behind Katy and say what happens to these corals at ecosystem scale. Which corals do get hardened, and how long does that resistance last? Does it only last 6 months, or does it last indefinitely? And we don't really know the answers to those questions yet, but we can test them right here at the Biosphere 2.
Thank you. One of our questions from Kendra was, what animals, if any, do you plan to add to the ocean biome? So maybe building off of stress hardening, starting with corals, but then going more broadly to other species of fish, other invertebrates. What is kind of the game plan for adding additional animals?
Yeah, so I will put this up as these are 4 of the species of corals that we have plans to bring to the Biosphere 2 in sort of the first set of experiments that we're doing in collaboration with the Hawaiian Institute of Marine Biology, so the institute that Ruth Gates was director of, led by Ty Roach in particular. And we are going to bring these corals and put them first in the raceway, experimental raceway, grow them, test various solutions, and transplant them into the Biosphere 2 ocean. Unfortunately, this was something that we were planning on doing in the coming weeks. And given the current situation, that's on hold a bit. So to be continued when that will happen. But nevertheless, we're cruising forward and are really excited to have a new postdoctoral fellow, Dr. Dan Killam, who is an expert in giant clams. And so Katie and Dan and I, we're gonna, over the coming weeks, get giant clams into the system and start growing them to try to understand how they respond to stress in the Biosphere 2 ocean. Because much like corals, giant clams actually also have this small photosynthetic algae that live in their tissue. So we can understand a lot of the same processes by studying giant clams.
Thank you. We have a couple of questions, one from Andrew and another from Bill, and they kind of blend together well. Andrew asks, have there been any unexpected effects of increasing CO2 in the ocean biome? How was carbon introduced and measured inside B2? And Bill asks, have we learned much about coral bleaching from this lab? And I thought maybe this would be a good time to talk about the origins of the B2 ocean research, particularly the Columbia days, what that experiment was, but also talk about it in modern terms with how we would measure carbon dioxide in the ocean, and if we were to initiate any kind of other increased stress event, how that would be initiated into the system today.
That's a great question. So the good news is technology has advanced a lot since Chris Langdon's seminal experiments. So in these really seminal experiments that were done in the late 90s, he actually meticulously added acids and bases to reproduce the target pH, so the amount of CO2 that has been taken up by the ocean and ultimately changed the acidity of the ocean. And he did this to sort of mimic what at the time we thought was going to be the pH of the ocean at the end of the century, so at the year 2100. But we actually now know, if we continue, here is us in 2018. Unfortunately, as you can see, we are on the sort of worst case scenario emissions trajectory. So we are continuing as business as usual. And on this sort of red worst case scenario trajectory, we'll actually get to this condition that Chris Langdon reproduced in about the year 2050, so about 50 years sooner than he expected. And as Katie said, he showed definitively for the first time that corals were growing as much as 40% slower under these conditions. So this was really important work, and Chris Langdon is still involved in this project. And now in 2020s, we have the technology to actually bubble in CO2 gas and directly change the pH of the ocean. And this allows us to keep a very steady pH, whereas his experiment was kind of varying around some average value that he was targeting. So we're really excited to reproduce this experiment and more importantly, test these solutions and which ones work under these changing ocean conditions. And this is not related to your question, but kind of shows the advance of technology related to temperature control. This is what the latest and greatest, what we call a heat exchanger, that controls temperature of the entire Biosphere 2 ocean, so 700,000 gallons, at a rate of up to 2 degrees Celsius, or about 3.6 degrees Fahrenheit every single day. So we can really rapidly change the temperature of the system to really stress out the ecosystem if we want to reproduce these types of really extreme events that we expect to see in the future. So for reference, compared to this latest and greatest plate and frame heat exchanger that's made out of titanium, state of the art, there was an entire room in the back behind where you can't see behind Katie, where the old heat exchanger was. And so we've come a long way in the last couple decades, and we have a lot of technology on our side to really do groundbreaking research on coral reef resilience.
We have a couple of questions, one from Doctress and another from Leslie, that talk about some of the unexpected kind of positives of the coronavirus pandemic in terms of a decrease in carbon dioxide emissions, a decrease in pollutants. As a geoscientist, how do you think that coral reefs might benefit from this situational pause that we're taking?
That's a really good question. And to be honest, it honestly matters whether it is a temporary blip in behavioral change, or if we take this moment to pause and reflect and realize as a community that we have some profound work to do. Because as we've seen, for example, following the 2008 crisis, that there was certainly a blip in the emissions, and people started buying less fuel-intensive vehicles, and there was certainly a lot of positive changes that came out of it. But unfortunately, we tend to resort back to our habits very quickly. And if we do that, we will still be on track to continue to be on this sort of worst case scenario. So I think there's a tremendous amount of hope for what's happening in terms of the climate impact it could have. But that will only matter if we continue to have these major behavioral changes and ultimately a worldwide political sort of agreement to reduce emissions. So I love the optimism, and I really sincerely hope that that is one of the things that comes out of this really tragic situation.
And we have a couple of questions from Facebook, one from Garrett, one from Heather, that talk about some specifics in coral reef. One is, how long does coral take to die after it gets bleached? Another is the coral reef an endangered species? So maybe you guys could talk a little bit about the process of bleaching, how long it takes a coral to die, and then are corals recognized under endangered species acts in our kind of modern system?
Yeah, so I mean, the short answer to the first question is it really depends, which is kind of a crappy answer, I'm sorry, but it depends on how long, how intense the warming lasts. So this example that we showed earlier in the 2014 to 2015 extreme event that hit a lot of reefs really hard, a lot of reef loss happened within the course of a season. So you can see here from December of 2014, there's a healthy coral reef where you can see that coloration that comes with the photosynthetic algae. And then bleaching, so the skeleton is left behind and the clear coral tissue, so that's why it gets its name. And then it's a little tough to see, but by August of 2015, these corals are actually covered with algae. So even though they look kind of brown in color, this is turf algae like what we have in the biosphere too. It's growing over the coral. But in less extreme events, or if there is a gap in between individual extremes, corals can recover. They can actually, like if there's a part of the colony that dies, they can actually translate energy and nutrients horizontally because they're colonial organisms and regrow. So they don't always die, and it really just depends on how the event plays out. And then there was a second question. Sorry.
Yeah. Endangered species, and are corals endangered?
So there are a few species of corals, particularly in the Caribbean, that have been put onto the endangered species list. Elkhorn coral being a classic example. This was I wish I had a picture in here, but it's very closely related. It's in the same genus as this picture here. But it looks like an elk's horn. So if you can imagine, it looks kind of like this. It's a beautiful species that used to be all over in the Caribbean, and now has been almost, entirely lost across the Caribbean reefs. So there are examples of corals that have been put on the endangered species list. And more broadly, corals as a whole are really at risk, particularly on the trajectory we're currently on in terms of climate change.
And we have a question from Kat. She says she knows that acidification causes calcification among soft-bodied marine organisms. Does this have a major impact on coral reefs? And perhaps you could talk a bit about any kind of mutualisms that exist between soft-bodied organisms and the reefs that they kind of are a part of.
So I'm not sure I totally understand the question. Acidification causes calcification among soft-bodied organisms. So, ocean acidification has a variety of impacts on corals of different types. It has the largest impact on hard corals. So the types of corals that I'm showing here that make this skeleton out of calcium carbonate, so that same mineral that that makes a shell on a beach. And that's because, like Katie said, that it impacts the availability of these key building blocks of their skeleton. Soft corals are certainly impacted in other ways. And to be honest, we are constantly learning about the complexities of various organisms' response to ocean acidification. And some have very complex relationships with this change. And so it's not always as straightforward of an impact as it is with these hard-bodied corals. But just to bring it back to why this is important, it's not that sort of soft-bodied corals aren't sort of as important for the reef, and obviously any impacts of ocean acidification on coral reef organisms is really critical. But the reason why we always focus so much on these hard-bodied corals is that they are really critical for these ecosystem services that reefs provide. So we often like to give this analogy between coral reefs, and this is something that Ruth Gates always did, and I love it, of comparing them to New York City. Like New York City, coral reefs are so biodiverse because of this complex 3D structure that these hard corals create, that produce these amazing nooks and crannies and habitats for reef dwellers. So just like our cities that have complex infrastructure that support these vibrant and growing populations, so do our coral reef organisms. And so really we focus a lot on understanding these hard-bodied corals because they are so critical for the biodiversity of the coral reef as a whole.
We have a question from Judy Andow. She's asking about the algae removal and if the U of A furlough and the coronavirus quarantine is an opportunity for people to get involved in continuing to remove algae, or perhaps more generally, we could talk a little bit about how people can get involved in the Biosphere 2 ocean research as a whole as well.
So yeah, in terms of diving, we definitely, people that are dive certified, we've had volunteers over the years that have joined our research team. We try to keep our groups going in the ocean at a minimum, because the more people we have in there, the more we stir up the sediment. We try to really make sure we're not stirring and disturbing that sediment, because we see an effect on our nutrients. So absolutely, if you're a certified diver, reach out to us. We have a list that we will occasionally reach out to if we have a big push. And in terms of right now, we don't have many people on site. We're practicing all the same protocols that everyone else is. Just a few of us are here making sure that the systems are still running. So right now, probably not the best time, but when we can get back to being together, that's definitely an option. And the best way to get involved is to reach out, if you're local, to reach out to us. We have volunteers that are part of the system that help us with daily tasks, but also just to follow along on social media platforms and our website. We have all these different ways to connect with us there and to follow along on the project.
And in the spirit of the current project, could both of you talk about what's to come in terms of adding infrastructure and continuing to develop the research here?
Yeah, absolutely. Obviously you saw that we've already come a long way in updating the infrastructure and have added a number of new organisms, as Katie described, to the system. So the next big step for us in terms of infrastructure is this other piece I didn't show here, which is this amazing truss of lights that can hold up to 288 lights to supplement the amount of light that reaches the Biosphere 2 ocean reef, because we know that it needs to be supplemented because of the space frame. And so that's going to be the next big project for us to tackle, and this will really ensure that we're able to build a really vibrant and healthy coral reef ecosystem in the Biosphere too. But a little bit more broadly, we are really thinking about this project in terms of this sort of three-phase structure. And we've talked a lot about phase one of this major project, which is where we've been in the past couple of years, which is really monitoring this degraded system to understand everything we can about how these degraded systems operate, how nutrients cycle, and things like that. Rejuvenate the system, so this is adding the key herbivores that Katie described, and then ultimately physically pulling the algae to clear the way for the coral phase of the experiment. And this is that bioremediation phase. So that's largely behind us now. We've finished phase one. We've learned a ton, and we are excited working up some papers to publish on our results from that part of the experiment. And now we're working on this sort of engineering phase of building a resilient reef in the Biosphere too, and starting to test these solutions for coral reef restoration. And so in the coming months, we'll start introducing corals. We talked about these giant clams that we'll bring in and start to build this diverse ecosystem. And then finally, in the third phase, in the coming years, once the engineering is completely in place, we will sort of turn up the knobs, if you will. We'll try to mimic what we expect the future ocean conditions to look like on these reefs, and test these solutions under climate change scenarios, and really ensure that we come up with solutions that will ensure the persistence of reefs for generations to come.
And we are reaching the end. We've got a couple more questions. One was about artificial wave action, which we did talk about earlier. Wouldn't it be helpful in Biosphere 2's reef? Well, the answer is we do have some artificial wave action generated by a wave generator on the back wall behind Katie.
You can't see it right now because I turned it off because it's pretty loud. So that's the reason the ocean looks so still.
Yeah.
It sounds like a like a dinosaur from Jurassic Park. And the other questions we have, which is an interesting one, is can coral reef be invasive? And perhaps you could talk about the idea of bringing in novel species into the Biosphere 2 ocean, and how that's both got its pro sides and con sides potentially.
Yeah, I mean, that's a great question. So to start with, the key piece of information here is that coral reefs are not all the same in terms of their response to climate change. So like many ecosystems, there are species that we consider weedy species. So even though they aren't invasive species per se, they can often dominate reefs following disturbance. And so you'll go to a reef where you'll see, actually I don't have a great example of that on this slide, but these sorts of branching coral species tend to grow really quickly and therefore often sort of take over, just like weeds in a forest, following these sorts of stress events. And so as Erin was alluding to, one of the big questions we want to look at is what sort of combination of species can create the most resilient reef that not only persists, but also still has these key ecosystem services. Because you can imagine, much like a field of weeds versus a forest, the services that those weeds provide is very different than that of the forest. And so we need this combination of diversity of species to give us that key societal benefit that reefs provide. And so the bottom line is we know reefs in the future will likely look different under stress, and so they might not be what they are today. But we need to balance having a diversity of species and being able to hit these key sort of functions that coral reefs provide, and make sure that we don't have just invasive types of weedy species take over the reef.
Diane and Katie, thank you so much for commemorating the 50th anniversary of Earth Day with us. And it's been a pleasure to have you on our last webinar of the day. The next event for Virtual Earth Day is a series of very special SARCEF presentations that you guys will all be able to find on our feed. And we appreciate all the listeners that we've had, and I can speak from my own experience that Katie and Diane and the team at Biosphere 2 in the Reef Lab are a group of very impassioned individuals that are really looking to solve problems in our oceans. So we're lucky to have a team like this with leaders like this. And thank you guys for tuning in today. We'll be back with more virtual events in the short future here. And you can follow all of our research on our social media, or look up more specifics at Diane Thompson's lab website. Any last words for our oceans and the Biosphere 2 oceans, Katie and Diane?
I would just add to, like Katie said, we are super excited to have people involved, especially from the local community. And even if you're not local and you're excited about what we're doing, please go to our website. We honestly couldn't do what we're doing without a tremendous group of collaborators and supporters. And so go to our website, and we would love to have your support if you guys are as excited about our solutions as we are. So we look forward to seeing you virtually or the next time at the Biosphere 2.
Thanks, everyone. Happy Earth Day.
Happy Earth Day. Take care.